My question goes to Dr. Hui Shen. This is really fantastic talk, very, very exciting, and fully take advantage of STOmics technology. So I actually have many questions, but my first question, maybe I didn't catch the main point. So you use a post-mortem brain tissue, right? And typically this is nucleus-based, or you are the whole tissue, because you detect mitochondrial gene expression. But in a typical single-cell RNA sequencing, which I don't know the STOmics, but in a typical single-cell RNA sequencing, we try to remove the mitochondrial gene, and to basically this is the contaminants we want to remove. But you use this to detect those kind of stress. So I want to know how you deal with it.
I think the technology part, I think that Yongfu can answer that. But for us, I think we use so we do detect the large number of the mitochondrial genes in our data. So whether the Stereo-seq actually excludes the mitochondrial gene, or Yongfu can answer.
Yeah, probably I can jump in a little bit. There's actually literature shows that the stress neurons, stress cells, they have higher proportions of mitochondrial genes. So and also it's related to cell death or apoptosis. So that's why in typical analysis, people exclude set a certain criteria for mitochondrial genes. If that proportion is high, then they probably exclude that from further analysis. But there's literature to support. Actually, this can.
My question is not to address whether mitochondrial gene is important or not. Obviously, biologically, this is very important. My question is more like a technical one, because when you do typical single-cell RNA sequencing, if you single nucleus, because post-mortem, you don't have fresh tissue, so you typically only detect the nucleus gene, not the cytoplasm gene. And mitochondria is in the cytoplasm, it's not in the nucleus. If your method is based on nucleus-based sequencing, and if you see high number of reads from mitochondrial genes, you have to consider this a high contamination. And typically we have a cutoff to filter out those short reads to avoid this kind of the noise. So that's my question. Whether if STOmics can do both, that's great. The neuroscience probably we can take a lot of new advancement. That's actually I'm very excited with.
Yeah, so for those data, we do use mitochondria readout as the quality control in general speaking, right? So you do want to have a better quality of your sample to go with. And the pipeline can definitely do that. We can check that as a basic QC matrix, and that's the first thing we do to look at those things. And of course, for Dr. Deng's researcher, I think they look at even deeper and just to see what kind of specific those genes shows up in the different locations. So that's not like generally like a single cell, right? So you have a contamination here, but there we have a spatial information. And then it's possible we could have, higher mitochondria readouts indicate more cell deaths or whatever, whether that's the sample quality or that's more like biological related. That's why you use the different module, different things to kind of test against with each other. But speaking of, for the technical speaking, Stereo-seq pipeline does provide the tools to look the QC. And also, if you really want to look at those deeper into the mitochondrial genes, you can still look at it. And we have those data there.
Yeah, that's very helpful. On the other hand, potentially, like Dr. Deng mentioned, this very strong evidence. And the mitochondrial gene, they are also nuclear gene can to regulate the mitochondrial gene. That's another way to see the stress in the specific neuronal cell type. That's also the way. Yeah.
Yeah.
But I really like your talk. It's a very exciting presentation. Yeah. Okay,
Thank you.
Go ahead. Yeah.
Thank you. So Stereo-seq does provide a lot of data So in our raw data, we have terabytes of this information. And by default, there's sort pipeline analyze maybe just a portion of those data. And the researcher actually needs to go dive into that raw data and search the mechanism. So in general, we do have other customer may look at the mitochondria gene portion number, they'll say, hey, this quality is not good. Right, we do have that. We do have heard that kind of comment. But when you really want to look at the difference, you test about the different model, you really rigorously test it using the different modules and different analyze across the real designed experiment to rule out other artifacts and then conclude this is really biologic related. That is, you can definitely look at the Stereo-seq, and Stereo-seq can actually provide that amount of the data for bioinformatics like yourself can look in. But this is a kind of bottleneck for many other researchers. They have this data, but they really struggled to find the true meaning. Yeah.
Hello. Yeah. Great talk. So I'm just wondering, like, you have the 2 severe and then 4 moderate samples. So do you know, like, the genotypes or any ApoE or even like rare mutations in like the severe versus the moderate that can explain some of these differences?
We haven't looked at it this time, but we definitely are doing this, more analysis on this sample. And in fact, we also have really recently got the HE staining for the A beta and tau. So we're looking into how to integrate that as well into our analysis.
Okay, thanks.
Yeah, my question is for Dr. Shen. Have you looked at other brain regions besides the prefrontal cortex?
So at this time, we for the Stereo-omics, we only did this Brodmann 10 region, but for we are also trying to study all the other brain area, and we have a study doing using a different platform doing the hippocampus area that is ongoing.
Yeah, I have many questions. They're really, really exciting. I'm sure this will be top journal publication. So maybe 2 short questions. The first one should be very simple. You mentioned that you have like 3 samples in each group, right? Did you see like which kind of variation among those samples in the 3 different groups, severe, moderate versus control? Did you see the strong variation, or it's pretty homogeneous?
You mean within each group?
Yeah, overall, you compare 6 layers, number 1, later mitochondrial, later you compare to gene modules. Did you see overall consistent pattern, or you see the variation among the individual is compatible with the variation among the groups, which is common, question we like to know.
Batch effect, right?
Yeah, but more where batch effect or sample. Yeah, batch effect covers 2. Biological batch effect and the technical batch effect. Yeah, right. Yeah, I'm asking more biological batch effect.
So for the severe AD group, we only have 2 samples basically. So there's 4 moderate and 6 controls. So for the network analysis, we actually basically combine to identify those modules. We combine all the sample together to identify those modules. So within each of the group, we probably don't have enough data to differentiate those.
So I can add on to that. We have a slightly different application, which we are not talking about yet, but we work with brain organoids that, and we sort of like embed the 6 brain organoids in one section. So we did a pseudo bulk analysis to compare, and across like 3 different sections, or 6 different sections, we saw like correlations of more than 0.95. So it's quite highly reproducible, I would say, despite like the sort of.
You actually mentioned another application. I think it's STOmics. It's the perfect technique for brain organoid or similar organoid because you can pretty much, manage the tissue growth and do this kind of
The
spatial
cell
transcriptome.
counts are very varied though. We get something like 6,000 cells from one slide all the way to 25,000 across another slide. But that's it. The overall like pseudo-bulk correlation is really, really high, like 0.95, really.
I can also add in a little bit on the technology reproducibility. I think Michael Snyder this noon, he also showed his data and between two replicates on the intestine samples, and the correlation is more than 98% sample to sample. And they also compare with single cell sequencing data. Yeah, the correlation is also above 95%. Yeah.
So the first question is single cell versus spatial transcriptomics. Is complementary or competitive?
Well, I think this one is more complementary, or when the technology improvement, they're probably gonna, basically merge into a single cell spatial transcriptomics, so that you can look at both at the same platform. So both are important, of course.
Yeah, like Dr. Shen mentioned that single cell now has or given much higher, not much higher, slightly higher capture efficiency. For example, PBMC, cell culture, they can have 1,000 genes, 2,000 genes per cell. And for any spatial technology right now, it's ranging from a couple of hundred genes to 1,000 genes per cell on average. So for now, it is more complementary, and people are using, if you have spatial data set, people will plug in single cell data set to get better cell type annotation, those kind of questions to answer, and then dive into more details of following biologic questions. But in the future, with improved technology, like Dr. Shen mentioned, we think eventually it's going to be merged into one technology. Yeah. And the spatial will be replacing single cell.
Okay, the second one is submitted by the people who signed up, but so what's Stereo-seq scalability, and depth? That's basically the question.
Yeah, I can answer that from the technical standpoint. They want to ask about the scalability. So right now we have one chip, different size, on one slide, right? So as a technician, how many slides you can handle per day, that really depends on your experience and your ambitious goal, right? So we usually see people do 3 to 4 chips, right, in 2 days. But we do provide automation system. So we do have an automation called Go Spatial, and that's going to be available next year. So in one run, you can handle 24 samples, and you will get 24 libraries for sequencing in one day. So that is how we think about scalability. So we're thinking about the reproducibility as well, because the manual handling, there always have some variations, right, during the operation. And then we think about this providing the extra capital equipment for benefits of your studies and your investment. You can scale it up, and also you can have a better reproducibility. Talking about the depths, I'm thinking about maybe they want to ask about the sequencing depths for Stereo-seq. This is really depends on the resolutions, and also depends on how many molecules you want to profile, right? So the higher resolution it is, the more so you're trying to profile, and definitely will require higher depths of the sequencing. Typically speaking, for the 1 centimeter by 1 centimeter Stereo-seq chip, you put coral sections of the mouse brain, and we do require you have 1 to 1.5 billion reads to be sequenced. So that is how deep we recommend you to do, simply because there has more molecules to be sequenced and has a higher resolution.
I also want to chime in one comment here. The scalability is also dependent on the simplicity of the workflow. So we have recently been upgrading our reagent kit to version 1.3, which sort of cut off the workflow. And now the entire workflow on the chip cuts down to only 5 hours. So that will also help us with the efficiency and to scale up more easily. Yeah.
I think one problem with scalability is the price. Compared with, that's why in our proposals or in larger scale studies, we tend to use 10x.
Yeah.
It's much cheaper.
Yeah.
But I think a distinct advantage of your platform is actually its capture area. That big, that's, that can answer a lot of questions, like 10x cannot answer.
Totally agree. Yeah.
Like the interactions, how it depends on the locations in the tissue for the same cell interaction, how that may change with different brain areas.
Right.
So on.
Yeah, I think the cost is largely dependent on the production scale. Once we have more users, the price, that's definitely going to drive down the price significantly, right? For now, we're still at the earlier stage of, this commercialization of this product. Yeah.
So a related question, but still, like the other thing said, it's a part of scalability. So you looks like you mentioned this there's several parts of the tissue section. My question basically, in addition, 1 times 1 centimeter, do you have different options? So your customer can choose, okay, for some tissue, I just need a more refined way. Then you have more deeps, right? But the other tissue maybe more to cover more tumor. You can look at more areas, see this kind of lineage of the progression of the disease. Do you have different options? Or what's the advantage, disadvantage of it?
Wonderful. Thank you for asking. As you can see, we have different size to choose for you to choose. 0.5 centimeter by 0.5 centimeter, 1 by 1, 1 by 2, 2 by 2, and 2 by 3, right? So that's our regular productions, and you can order. And then we're going to have a customized size for researcher to order. It's 3 by 5, 4 by 6, right? So in the U.S. region, so we have this 2 size for researcher to customize and order it. So speaking of, putting the bigger tissue on the bigger chip. And for sequencing depths, you don't have to do the deep sequencing at the first run, right? So you can do a shadow sequencing and then see the quality and see if you can find some kind of preliminary insight. And then you can add more sequencing, right? And you can add even more later on. It's really flexible as long as you have the library, right? Did I answer your question?
Oh, yeah. That's a perfect answer. And I really like your strategy. This really, in reality, I'm running sequencing code. This really help the customer. Yeah, I like it.
Next one. How can spatial transcriptomics be effectively performed?
I think that's also from me. Okay, I'm a technical person. I can speak for that. For effectively, Stereo-seq spatial transcriptomics does require skill, right? For you to do it, for us to do it, for anybody in your lab to do it. You have to understand the tissue itself. And of course, you have to understand the biological question you're going to answer. This is a big investment. You better to have a good scope in first hand and really understand what you really want to look at, right? So, and then you can design your every single step, because the tissue preparation itself can affect the outcome, right? So if your tissue has been damaged, there has too long ischemia time, and of course your molecule has been degraded. And all this information add together will, sometimes you get the data you want, but sometimes you just don't get the data you want. So technically speaking, there has a multiple step in between. So you prepare the sample, you're sectioning and imaging and staining, right? So, and also pipetting even. So not myself, I may not be a good pipetter, right? So adding those accuracy, amount of the fluid is very key for this kind of molecular experiment. With that being said, any scientist can adapt this technology, because this skill is learnable. and we provide the training to the scientists to run the first time. And we can help you to adapt this technology and help you to operate in your lab. I mean, in terms of the efficiency, and you want to give yourself a little bit learning curve, and then you just learn each single step, and then you scale it up, right? You run first chip, and you run second, and you run third all together, and you can run it daily, right? So this is really depends on your goal. And speaking about the efficiency, that's just based on the practice.
I think I'm gonna speak from the product dev point of view. So we're also, like I said, we're trying to simplify the process for you to, learn the learning curve to be shorter. And also we're trying to, we will release this automation instrument next year. That will also help a lot with core labs or service providers. Yeah.
And that's Go Spatial, right?
Yeah,
Yep.
it is called Go
That's coming
Spatial.
next year.
And we already have one at San Jose lab, and our demo lab people loved it. It's very reproducible, and it actually also controls the diffusion, because one of the main problems you may have in your data set is for this unbiased whole transcriptomics capturing. There's always a little bit diffusion, right? So it is a balance between high capture rate versus the diffusion. Yeah, for automation, it actually minimizes the diffusion quite significantly.
Okay, should we move on to the next? How to analyze data at the single cell level?
AI. I answered the question. AI.
Oh, AI?
Only is the
train AI first.
First, here many experts how to do the single cell analysis and to look at the clustering across the tissue segmentation. This is one like standard or evolving technique, right? But how to refine, make it even more informative. So the deep learning algorithm. This is thing about imaging. Imaging has been very widely applied by AI technique, and similarly, this is the same thing. And I have read some paper or some other proposal, basically how to address this kind of, the more like a niche kind of neighboring clustering features. But I think we have a lot of fun here. I'm not an expert, you are. Yeah.
Yeah, so I mean, this is an unbiased, single cell spatial RNA-seq technology, unlike some of the more targeted questions. So I totally understand, right? A lot of molecular biologists who might not have the skill sets or the tools to be able to ask deeper questions beyond just standard clustering, they may be opposed to that, but at the same time, it brings out opportunities. So for instance, our lab works a lot with viral infections, and without an unbiased approach, it's hard for us to know what viral transcripts to design probes for and everything, so we are able to get really interesting and exciting host viral transcript co-expression from Stereo-seq, but not a more targeted approach. But again, it's very early days, and the tools have to be customized and everything.
We move on to the last one. Why is species agnostic important? Yongfu?
Yeah, I can, I may not be the best person to answer this, but as an ordinary scientist by training, I would say we have so many animals or plants in the world, right? So human health is very important, no doubt on that. But understanding all those fundamental questions, we cannot harvest the tissue all the time from a patient. You would have to based on some other animals or models, and you would rather to find the deep mechanisms through the evolutionary, right? So with that being said, you want to have a technology that you can work with different type of the organism. And for now, there have other targeted approach. Then you have to design the probes and design the things specifically targeting that species, right? Think about how many platforms you want to adapt to your lab or to your core. people may want to work with human, mouse, zebrafish, organoid, plant, right? So you, as a core or as a scientist, you probably have enough fund to get a mating technology, but not all. And for Stereo-seq, our inventor, when they are designing their technology, they want this technology to be widely applicable. And also think about the flexibility, right? So they design the different chip size for you to work on the different size of the sample, and also without suffering the high resolution. And also they keep this species agnostic capability in mind from the ground. Even they work with FAP platform, right? So they use the concept, still work with any species. So with that being said, I think this technology cover the majority applications in the single lab or in a core facility.
Question about the automation instrument going to be released next year. So what's the starting point and what's the kind of end points for that system?
Starting point, are you referring to the workflow?
You are referring to which step you put your material onto the automation.
Because the instrument doesn't integrate with a cryostat or a microscope, so it will be starting right after imaging steps, and then the next following steps during the in situ, for example, or on chip reverse transcription, everything will be with the instrument. And then it can go all the way to the before library prep. And then we'll also have a small automation because that has a, it can be also applied to other NGS library prep. So we call it SP100. So that small instrument will help you with the library prep part. So it goes all the way from imaging to library prep. It can be optimized.
So right before the library prep type step.
That's a pinpoint. I guess I'm answering your second question. So the pinpoint is that, unfortunately, if we're going to build everything into one instrument, so that's going to have the cross-section step as well as the imaging. So it's going to be a very, very large instrument.
Yeah. It's definitely understandable.
Yeah.
Library prep itself is not a pinpoint to a core, right? So we know how to make libraries. So
you don't need the SP100 if you,
Right. That's great.
But again, automation is not just for scalability, but also for reproducibility. And as I just mentioned, it can also minimize the diffusion problems if it's handled by human. Yeah.
Another point is for your investment, right? As a core facility, the most valuable investment is humans' time, right? So you are hiring more technician or scientists, or you can give the machine to do the work. So from the beginning, you may have to buy a capital equipment, but if you run this experiment with the 2 years or 3 years, you can definitely get that investment back quicker if you're using the automation compared to hiring more skillful scientists.
Okay, thank you. Thank you for 4 speakers, Yongfu, Dr. Shen, Dr. Lim, and Yuan. And thank you very much for you guys staying.